Okay, so as in the last video we have discussed about the RAS, which was renin angiotensin aldosterone system. In today's video we'll be answering two questions, two most important questions. So what those questions are, basically how renin is released, the first question, and the second question, how aldosterone makes urine formation down. Alright, so I am writing here the first question. Here I have first question, which is how renin is produced. All right, or how renin is released. This is single end renin, which is released by the kidney. All right, is released. So how renin is released? So we have to answer this question first. So I am going to make here the structure of nephron. So this is basically PCT. Alright, this is PCT, and here I am making arteriole, which is efferent arteriole. Then it is divided into capillaries for the filtration. Then again it is forming efferent arteriole, and here it is subdivided into capillaries. Alright, that I'll be making after. Then here we have what? Here we have PCT. Here we have loop of Henle, and then here we have DCT. So same loop of Henle and DCT. Alright, this is DCT. And so here in the women's capsule we have the cells which are like squamous epithelial, and here we have modified version of squamous epithelial. Those are known as podocytes. So here we have podocytes. These are all podocytes. Alright, these are squamous epithelial. In the PCT, we do have cells which are known as brush border cuboidal epithelial. Alright, here we have, wait, I should make this all. These are squamous epithelial, women's capsule. So these are all squamous epithelial. Alright, here we would be having filtration. Alright, here we'll be having filtrate. Okay, so here now it will go to the PCT, and in PCT we do have cells which are known as brush border cuboidal epithelial. So these are what? These are brush border cuboidal epithelial. In whole PCT these cells are present. Brush border cuboidal epithelial. I'll be writing this as well. These are all brush border cuboidal epithelial. Alright, these are brush border cuboidal epithelial. So now we have loop of Henle. In loop of Henle we have squamous epithelial cells. Alright, in loop of Henle we have squamous epithelial cells. So these are squamous epithelial. Wait. In the ascending loop as well as in the descending loop, squamous epithelial. These are the squamous epithelial. In the loop of Henle. Alright, so in the DCT we do have cells which are cuboidal epithelial, but here in the starting of distal convoluted tubule these cuboidal epithelials are somehow modified. So these modified cells are known as what? These are known as macula densa. So let me make here macula densa. These are like big one. Alright, these are macula densa and these are the modified version. Then again here we have simple cuboidal epithelium. Here we have modified version. Again here we have simple cuboidal epithelial. Alright, these are the cells of nephron. So let me write here as well so that it can be clear to you. In the Bowman's capsule, we have cells which are squamous epithelial, but modified version of squamous epithelial is podocyte, squamous epithelial. Alright, these are modified there, and that modified cells are known as podocytes. So here I am writing this as well. These are podocytes. These are podocytes. Alright, in the PCT we do have cells which are known as brush border cuboidal epithelial, brush border cuboidal epithelial. Alright, then in the loop of Henle we do have cells which are known as squamous epithelial. So these cells are squamous epithelial. Squamous epithelial. Alright, so in the DCT we do have cells which are known as macula densa. These are the cuboidal epithelial, but these are the modified version of cuboidal epithelial. So these are known as macula densa, while these cells are known as cuboidal epithelial. Cuboidal epithelial. Alright, so here I have mentioned their names as well. So now what we need to do, now we know that blood pressure is lower in this case. Alright, BP, wait, BP is low, and when BP was low, then kidney was like producing or releasing renin. Alright, so here in this case BP is low. So BP is low when this filtrate is here. It is here, wait, filtrate is in the Bowman's capsule. Alright, so it will go to the PCT, and in the PCT we know that the reabsorption of water is happening. Alright, so here I am making the capillaries, peritubular capillaries. So these are peritubular capillaries. I should write this as well. Peritubular capillaries. Alright, so here the reabsorption of water is happening. Reabsorption of water is happening. Alright, from the PCT. So here I should write 88% of the water is reabsorbed, and sodium ions are reabsorbed as well. Sodium ions. Alright, when sodium ions are reabsorbed, means the salts are now reabsorbed. Alright, so now when this filtrate will go to the DCT, first of all it will go to the loop of Henle. Then from the loop of Henle it will go to the DCT. So now this filtrate is tasteless. We know that, tasteless, because salts are already absorbed. So now this is tasteless. Now it has not any taste. This is tasteless. So now this will go to the DCT, and now DCT. Now DCT has the modified cells of cuboidal epithelial which are known as macula densa. Macula densa has the power of testing this fluid. So when it will test, and it will tell to the peritubular capillaries, it will inform these peritubular capillaries. So let me make these peritubular capillaries more big. Alright, so this will tell this thing to the paratubular capillaries. And then this paratubular capillaries, now I have to make this diagram again so that it can be more clear to you. Because we know that this nephron is not like this nephron is never like this. The Bowman's capsule is on the other side. I'll be telling you that as well. So here the secretion will be happening. Now I'm going to make another diagram of the nephron secretion. All right, so I am, first of all I should write here, this is PCT, then this is loop of Henle. This is DCT, which is distal convoluted tubule. Wait. All right. And this was the Bowman's capsule. Bowman's capsule. I'm not writing that. All right. This is afferent arteriole, and then this is efferent arteriole, while this is glomerulus. Alright, so here I have labeled each and everything. Now I am going to make the structure of nephron again, and that would be the cleared structure. Alright, now I am going to tell you how in our book the nephron structure is given. So Bowman's capsule is placed like this. This is basically Bowman's capsule. And from, wait, this is basically Bowman's capsule in our book. It's like this. Then from there PCT is coming. Then PCT is like this. Then loop of Henle is going down. This is loop of Henle. Then this is DCT. And from here we have collecting duct. So nephron structure in our book is like that. And you know that the artery is carrying blood. So this is what? This is the afferent arteriole. Then it is subdividing into capillaries. And then this we have efferent arteriole, and then it will divide into other capillaries which are known as paratubular capillaries. So here, as I told you, macula densa cells are the cells which have ability to test this fluid, and now that fluid was testless. This fluid was this fluid was testless. So this fluid is testless. When this tested, and it has the cells which are known as juxtamedullary cells, juxtaglomerular cells. Sorry. So here it has cells, small cells on the macula densa, and these are known as what? These are known as juxtaglomerular cells. Juxtaglomerular cells. Alright, it has cells which are known as juxtaglomerular cells. So now these juxtaglomerular cells, and here in the artery we know that we have here smooth muscles on the efferent arteriole. Alright, on the efferent arteriole we do have cells here which are known as smooth muscles. So here I should write as well. These are smooth muscles, and these are the cells of afferent arteriole. Alright, so juxtaglomerular cells will give signal to the smooth muscles, because they are like very near to each other. Juxtaglomerular cells are very near to the smooth muscles, so it will give signal that the fluid is testless. So the fluid is testless, so smooth muscles will start producing renin. Now they'll start producing renin. Here they'll start producing renin. Alright, they will start producing renin, and then again it will go to the filtrate. Wait, here would be the filtrate, as we know that the filtration is happening. So here we have filtrate now, and it will go to the PCT, then it will go to the loop of Henle, then again it will be absorbed here from the paratubular capillaries, and then paratubular capillaries, we know that, are forming what? They are forming vein. From here, all things, like from the vasa recta as well, these all paratubular capillaries are forming here what? As we talked about this in the structure of nephron as well, this is forming vein then. So that vein is carrying what? That vein is carrying renin with that. Renin with that. And renin is then came to the blood, and then again renin will combine to the angiotensin which was released by the liver, then it will make the blood pressure higher. So it will, renin will be carried to the blood and it will be out of the kidney. So this is how renin is produced. Just because the blood pressure is getting low and this is filtrate is getting testless, then that's why macular densa are giving signal to the smooth muscles of the afferent arteriole, and smooth muscles are producing their secretion which is known as renin, and renin is this then carried by the vein to the blood. All right, so this is how renin is produced. We have answered our first question. All right, we have answered our first question. Now we have to go towards the second question. All right, and what is second question? Second question is how aldosterone makes urine formation lower, because we know that the aldosterone was a hormone which was released from the adrenal cortex, which was placed on the up of the on the top of the kidney. It was produced by the adrenal corner, and it acts on the cells of DCT same, and then it increases sodium potassium channels in the DCT. All right, so we are going to make here, I am going to make here the DCT. All right, so now we have the second question. Again we have second question here. So second question is what? How aldosterone makes urine formation lower. Formation lower, down or lower. Alright, so this is the second question which we have to answer now. As we know that aldosterone is a hormone which is released from the adrenal cortex, so it will act on the DCT cells. So here now I am going to make the DCT. Alright, so this is the basically DCT. This is what? Distal convoluted tubule, which is also known as DCT. Alright, this is DCT, and DCT has cells, cuboidal epithelial, which are modified version of cuboidal epithelial, and those are known as macula densa. So here I am going to make those cuboidal epithelial. Alright, these are what? These are cells of DCT. Cells of DCT. Alright, so now what will be happening? Wait, let me make here the paratubular capillaries. So here we have paratubular capillary. I'm just making one paratubular capillary. Paratubular capillary. Alright, this is paratubular capillary. Now what will be happening? Alright, we know that the absorption of the water is like water is absorbed from the reabsorbed from the DCT as well. Alright, so here aldosterone has the power to increase the pumps or channels, sodium potassium pumps in the cells of the DCT. We know that in the cells we do have sodium potassium pumps or channel. So what it will do, this aldosterone will increase the sodium potassium pumps here in the cells of DCT. So these are all increased, and these are all what? These are all sodium potassium pumps in the cells of DCTs. So let me write this as well. These are sodium potassium pumps. Alright, aldosterone has the power to increase the sodium potassium pumps in the cells of the DCT. So when these sodium potassium pumps will be increased in the DCT, then here from here the water, and here we do have what? Here we do have sodium ions. Alright, we know that these pumps will utilize 1 ATP, and 3 ions of sodium will be going to the paratubular capillaries, and from the paratubular capillary 2 potassium ions would be secreted to the DCT. So here, as there are so many sodium potassium pumps in the cells of DCT, so there would be so much absorption of this sodium ions. Alright, sodium ions, and from here the potassium, 2 potassium ions would be secreted. Alright, so I'm making this with white color. Alright, and if sodium is going there, as we know that sodium behaves as magnet. So if salt is going there, then water will go with it as well. Alright, water will go with it as well. So just because of this, this reabsorption of the water and sodium ions to the paratubular capillaries, it will increase the blood pressure. It will increase the blood pressure, and urine formation will be low. Urine formation would be low then. Alright, so this is how aldosterone makes the urine formation low. So now if BP gets high, now we know that BP is high just because of renin and aldosterone. BP is high now. Alright, I should write this as well. Blood pressure is high now. Alright, if blood pressure is high, then heart has a problem now. Heart has a problem with that. Heart has a problem. Now what heart will do to decrease the BP again? Heart, wait, let me make here the heart. So this is the heart, and here we know that heart is divided into 4 chambers. The upper are known as atrial and the lower are known as ventricle. So these atrial will produce a factor. They'll produce one factor which is known as atrial natriuretic factor. Alright, which is also known as ANF. It is released by the atrium of the heart, which is known as atrial natriuretic factor, which is the enemy of RAS. It is the enemy of RAS, which will do what? Which will decrease the blood pressure. Decrease BP. How? Just because it is a powerful vasodilator. Now we know that vasoconstrictor was what? Vasoconstrictor was angiotensin 2, and here vasodilator is atrial natriuretic factor. So here I should write this as well. It is a vasodilator. Dilator. And we know that volume is inversely proportional to the pressure. If volume is low, like is volume is here, it is increasing the volume of the artery. Vasodilator. Dilator means it is increasing the volume. So if volume is increased, then the pressure would be decreased, because we know that these are inversely proportional to each other. So vessel volume is increased now, and pressure will be decreased. So pressure would be decreased. This is how it is a vasodilator, and it is making the blood pressure low then. All right. So this was all for today, as we discussed about two questions: how renin is released and how aldosterone makes urine formation. Then we discussed this as well: if blood pressure will be high, then heart is a problem, and heart will release a factor which is arterial natriuretic factor. So we have discussed all of these. So in the next video we'll be discussing about osmolarity. The osmolarity, will be discussing about that as well in the interstitial fluid. So this was all for today.